after the ligand hit, we wouldn't be able to regulate the pathway. So what I mean by that is regulation is starting, stopping, speeding it up, slowing it down. You wouldn't be able to amplify the signal. You wouldn't be able to have other signals start different signal transduction pathways, which happen quite a bit. And you wouldn't be able to have layers of control each step along the way, which would allow you to tailor the pathway and the response. So it has to be complex to regulate it and to allow it to start other pathways. And if it wasn't, it would just be a simple, you're making 1 million of these proteins or you're making 0. But there's sometimes when you need somewhere in between. So that's why it has to be so complex.
Will you explain a couple symptoms of diabetes and like why they happen?
Like which ones?
Well, I remember yesterday in the video it was talking about like ketoacidosis. So
Yeah.
like, could you explain that a little more?
Yeah, sure. So here's the deal. If you have diabetes, your cells don't have the ability to use glucose. We can't get glucose into the cells to make energy. So in response to that, your body is going to use other items as energy sources. The other two items that your body would use would be proteins and breaking them down into different components, and also fats and breaking those down into different components. Both of those, when you break them down, some proteins, all fats, produce an excessive amount of, they're called keto acids. They're keto acids. And these are acids that donate H+ ions in the bloodstream as they're traveling around and getting to other cells so the cells can use those as energy. So the problem with that is if you have many keto acids in your bloodstream, they're acids, which is going to decrease the pH of your bloodstream. Unfortunately, when you decrease the pH of your bloodstream, it starts to interact with many other things. Number one, it's going to dehydrate you because more water is going to go into your bladder and get out of here. And you're also, because of the way things work in different cells and in the kidney, you're going to be depleted of K+ ions as well. So some of the symptoms are because of the acidosis. You have a low pH in your blood, lower than what you should. And because of that low pH, you don't have K+ ions where you need them. What are the places in your body that need K+ ions? Your muscles, your nerves. They help you send impulses. So you're going to have an issue with sensations of feeling in those nerves. You're going to have problems thinking sometimes. You're also going to be fatigued because your muscles aren't going to have enough energy to do what they normally do. And they're also not going to have the ability to conduct impulses because of the K+ problem. The other thing that starts happening is that you start breathing really fast, because breathing fast gets rid of carbon dioxide, which helps increase the pH in your blood. So all of those things are kind of dehydration. That's because glucose and K+ ions are going to be leaving in your bladder. So you're dehydrated. And then all those other things with the pH changing the K+ ions affects nerves, muscles, and all that other stuff. So yeah, ketoacidosis is not good. Somebody put something in the chat.
How does a transcription factor affect RNA polymerase?
Yeah. Okay. Was that you, Sahana?
Yeah.
Okay. Okay. So here's what I'm going to do. I'm going to do this visually for you because I just feel like it's the best way to do it. Inside all your cells, every single cell that you have in your body has 46 strands of DNA. And it contains every gene that you might want to turn on. Now, we realize it would be stupid and energetically idiotic if in every cell you always made every protein all the time. So we need a way to control what genes are making proteins. So we need a way to turn those genes on. So if I told you that this on this chromosome, and that just represents a chromosome, this is gene 1. This chromosome, we'll call this gene 2, and this chromosome is gene 3. And all of these are strands of DNA that we would find in the nucleus. Well, who else is in the nucleus all the time? This enzyme. And this enzyme's name is RNA polymerase. RNA polymerase is not going to start to transcribe every gene as shown, because it's not going to be able to find it, and it's not going to be able to attach to the DNA when it looks like this, which is a good thing. Because if it could attach and start to transcribe, we would have every stinking protein made that all 46 of our chromosomes have on them. And that would be terrible. So what we have is a system where we have a transcription factor that has to attach first. Now, a transcription factor could be just a phosphorylated kinase at the end of a pathway. It also could just be an intracellular receptor, which is a different protein. In the end, all transcription factors do is the same process. They're going to attach to certain places on your genome. In this scenario, I'll say that it's going to attach here. When a transcription factor attaches, that is the only time that RNA polymerase can find and attach to the DNA. Now, once this attaches to your DNA, it is going to transcribe the gene, which would be gene 2. Transcription is turning DNA, and this is DNA. It's going to turn this gene 2 area into messenger RNA, which is single-stranded. It looks like this, and here are the different things. So it's going to copy the instructions on one side of the gene, turn it into RNA. When we turn DNA into RNA, that is called transcription. And the only way that we transcribe, which is the purpose of RNA polymerase, is if a transcription factor is attached next to the gene that we want to turn on. So this is how we turn on specific genes. This is why specialized cells only have certain genes turned on, because they have pathways that end up in transcription factors that only will attach to certain genes and therefore only make certain proteins. And that's why all of our cells are so special. Just like Joshua. So that's the purpose of a transcription factor, and specific transcription factors that will attach to specific regions of your genome or DNA. Go ahead, Holden. Was that a wave goodbye, or is that a question?
No, I have a question. So a couple of days ago, you talked about calcium as a secondary messenger. And then in our proteins, like the first week of this unit, there was that ion gate. And the ligand opens up that ion gate, letting sodium in and potassium in. Is sodium and potassium secondary messengers?
They are not. That's an excellent question, Holden. Sodium and potassium are ways to set up electrochemical gradients. Eventually, we'll learn that muscles and nerves use electricity to conduct information, not just proteins moving, which is very different. So that allows them to set up the gradient. Now, there are calcium-gated channel proteins. And these calcium-gated channel proteins are what have to be open to allow calcium to exit or enter the cell cytoplasm. So we have pumps that pump out calcium or pump in calcium depending on the cell. And then we have gated channel proteins that allow calcium to move in or not. But calcium is a very common secondary messenger. For instance, the one I gave you was muscle cells, right? And muscle cells allow calcium to move into the cytoplasm and interact with actin and myosin, you could contract. Calcium also is the ion that causes nerves to release neurotransmitters. And that's a different secondary pathway that's utilized by cells using calcium. The reason why I gave you the skeletal muscle one is just to kind of give you an idea that the reason why calcium is used in muscles is because it's so full of proteins. There's not enough space for a kinase cascade to work in a muscle cell. They're just too big and fat for them to be effective in that kind of cell. That was a good question.
What are the two different general responses that occur in a cell due to signal transduction pathway?
What are two general responses? Oh, yeah. Yes, Sahana. Who typed that? Was that you, Sahana, again? Yes, I'll do whatever you told me to do after I answer short and long distance. Okay. So in general, you're either going to activate a protein or an enzyme. And then, like, an example of that would be muscle contraction, right? You're just activating actin and myosin so they can interact and contract, okay? The other possibility is you're going to turn a gene on, which involves a transcription factor. And then once you turn a gene on, that's going to create a protein. So one of the questions that I might ask you would be long-term response versus a short-term response. Like a short-term response would be muscle contraction. Boom, you activate a protein, you deactivate it, it's done. That happens quickly. If you're going to turn a gene on, you're going to make a protein, and that protein is going to do things usually for a longer period of time in the cell. So those are the two. They're both involving a protein in the end. It's just one just activates that protein or an enzyme immediately. Like you could activate an enzyme and do a process and then stop. And the other one is creating a transcription factor that's going to turn on a specific gene. So Hannah, the difference between long-term communication and short distance communication, the easiest short distance communication would be a nerve and a muscle, or a nerve and a nerve. Now, we haven't done anything with nerves and muscle, but if I was looking at a nerve and a muscle, it would be like, this is the end of the neuron, and the muscle is like right next to it. We're talking nanometers away. They communicate in very narrow gaps called synapses that allow them to communicate and do things. So that's about as small of a distance as you can move. And those two cells have to be really close to each other for them to communicate. Long distance communication is an endocrine hormone system where you have some type of gland that dumps a hormone into the bloodstream, and then the bloodstream travels everywhere in your body. For instance, the pancreas, when it dumps insulin into the blood, well, it's gonna allow a cell in your earlobe to let glucose in. And that's really far away from the pancreas. But how does it get there? Through the bloodstream. So those are two cells that are very far away from each other that still are interacting. And that's called long distance communication. Another short distance communication are immune cells. They have to be right next to each other and touch to activate. I love staring at all of you and awkward silence. I love it.
you just run through the signal transduction pathway for insulin in the case study?
Sure. Oh, shh. Sorry, I hit the wrong thing. Okay, so Avani, if you look at number 1, what kind of receptor protein system does that look like?
Receptor tyrosine kinase.
That's right. So those are the Wonder Twins. So we have insulin on each side. They come together, they activate, and then one of the twins is going to phosphorylate the other side. Now, this shows that we're phosphorylating in number 3 a bunch of different things. So if you notice, do you see we're phosphorylating this guy that says TF, and this TF is traveling into the nucleus. What do you think TF stands for?
Transcription factor.
There you go. Now, it also is activating this blue guy that activates a secondary messenger that turns on a different transcription factor that goes into the nucleus that's going to activate turning on a different gene. Okay, so we're activating multiple genes in this pathway using 2 different transcription factors, both of which that somehow are activated by these tyrosine kinase phosphorylated tyrosines. Now, the other thing that's going to get activated, we are going to initiate another secondary or transduction pathway that we have MEK, MAPK, ERK, and then it ends up in another TF. Holy mackerel, whatever that is. So this ends in K, this ends in K, and that ends in K because each of these is a kinase. MEK phosphorylates this guy, which then phosphorylates this guy, which then phosphorylates the ELK. And when you phosphorylate the ELK, it's an activated transcription factor that also goes into the nucleus. So we're now activating 3 different genes. We also are activating a system of these proteins being added to a vesicle. Now, all of these messenger pathways, one of them is initiating the creation and the movement of this vesicle, which is called GLU4. And the GLU4 travels up here. Exocytosis allows this to happen. When exocytosis occurs, it's going to move the vesicle up to this membrane, and then we're going to add these glucose transport proteins to the membrane. All of that, the creation of more vesicles, the creation of more proteins. If I want to create more proteins, I have to turn those genes on and make even more proteins. So that's what these other transcription factors are doing. They're creating more proteins. They're allowing the cell to create more vesicles so that when the time is right, we'll have lots of vesicles that are going to be released on here, and we can even let more glucose in when the time is right.
And then why does that one receptor have like so many different pathways?
That's the beauty of the receptor tyrosine kinase, is that when you activate that one receptor protein, it can activate multiple different pathways. So in the end, you can actually activate multiple genes that are involved in the whole process. So that's why it's good to be complex, because now we can initiate multiple genes being turned on, all with one signal starting it.
So is it just more efficient?
Yes, because if we if we didn't have that turning on all three of those genes in the nucleus, then we would need 3 different hormones to create the same effect that this one hormone is doing.
Why do some tissues that have like the same amount of receptor, why can they respond in different ways to insulin?
This is the Avani show.
No one else is asking.
I know. I love the awkwardness. At least give it 30 seconds. It's so much fun. Oh, wow. You know why nobody else is talking? Anant left. There you go. And you're not like at decibel levels that like destroy my eardrum. That's fantastic. Thank you. So I'm going to give you 3 different cells, and they're cells that I feel as though that are kind of important in the insulin realm. And I'm going to call the first one a normal cell. That's every cell in your body. And then I'm going to specify the liver cell and a fat cell. Sorry, I'll make it the gross green. It's easier to see. So in a normal cell, we just went through it. At the end of the day, we're going to do that whole signal transduction pathway that's in the case study. And that's going to let glucose into the cell. And the whole purpose of that, we know the whole purpose of glucose is to make ATP so we can run everything in that cell. Okay? So all cells do that no matter what. I'm going to give you 2 other cells that do something a little different. Now, I'm going to circle this, and I'm going to tell you, same. It does that too. And then I'm going to circle this. I'm going to tell you the fat cell does same. So they all need ATP. We know all cells need ATP, but the liver cell is going to do an extra special different pathway, and I'll just call it pathway 2. Pathway 2 is going to activate an enzyme that links together glucose into glycogen. Now, unfortunately, you didn't get to dissect the pig, but if you did, when we rip open the pig, the liver is huge. The liver is also a huge organ in your body as well. There's so many cells, it makes it a very nice organ to store things. Now, glycogen is a polysaccharide that your body uses as a storage molecule so that if all of a sudden it goes through a period of time where you're not eating food, you can start breaking down glycogen and turn it into glucose. That's what glucagon acts on. Insulin causes the creation of glycogen and the storage of that excess glucose that's floating around in your bloodstream. Okay? What a fat cell will do, they're not going to store it as glycogen. Fat cells store, so pathway 2, we're going to store extra glucose, modify it, and turn it into a triglyceride. Now, this is what a fat cell is called an adipocyte looks like. Here's the nucleus, and the rest of this is just fat. So our goal when we bring in that extra glucose is to store it as excess fat. And then if this cell gets a certain size, a different signal will tell that cell to divide and make another one, which is another fat cell. This is how we get fat, by eating too much. And then if those two are, then we'll just keep on dividing and making more if this happens. So insulin is linked to making fat cells because insulin causes your body to store excess glucose as triglycerides. So that's just a different pathway that is in the fat cell, a different pathway that's in the liver cell. Now, if you're going to run a marathon, your body will use all of the normal glucose that's in it. Then it will go to the glycogen. And after 20 minutes of running or jogging, like you would do in a soccer game, after 20 minutes, you just depleted all of your glycogen stores. Then after 20 minutes, your body goes to these cells and it starts using fat. That's why it takes 20 minutes of aerobic exercise before you start burning fat. And that's why normally people that run around a lot are not fat. Fun fact. But you could be a sprinter and still have some fat because they don't do anything for 20 minutes. I mean, they're lazy. If anybody here is a sprinter on the track team, I apologize. Oh, I forgot, Henry. You're a sprinter swimmer, aren't you? Oh, I thought you were 50. Oh, mid-distance. So you're good. You're good. So those are three different cells that need three different pathways that are linked to insulin, but a little bit different just because they're specialized cells, and you wouldn't want to store fat in a cell in your small intestine. It would be terrible. Oh, glucagon target cells. Glucagon target cell is a liver cell called a hepatocyte. And when glucagon attaches to it, it initiates a signal transduction pathway that activates an enzyme that breaks down glycogen into glucose and releases it into the bloodstream. So the reason why our body is making glucagon is because we have low blood sugar levels. So we need to increase the amount of sugar in our blood. So in response to that, our pancreas releases glucagon. And when the pancreas releases glucagon, it interacts with liver cells, which then rip apart glycogen into glucose and release it. So insulin resistance is related to, we do think it has a genetic component, but what I'd like you to think about is what causes insulin resistance is that in a human that makes a shit ton of insulin all the time, and that could be genetic, you know, your body releasing insulin more than the average bear, or it could be a component of eating bad food for you that's rich in carbohydrate, which is going to release more insulin a lot of the time. Or it also could be linked to not exercising as much, because then you're going to have more glucose in your bloodstream usually, which means more insulin. So if you have a lot of insulin interacting with cells all the time, eventually the cells are like, why the hell is this insulin thing interacting with them all the time? I'm getting sick and tired of that. It's like somebody tapping you on the shoulder all the time. And eventually you're just like, forget it, I'm going to move elsewhere. So those cells actually stop making receptor proteins and they decrease the amount of receptor proteins. And then eventually it might get to a spot where they don't even make the receptor proteins and then they're resistant. They don't react at all to insulin anymore. And usually that's over years and years of overproduction of insulin, depending if it's genetic or if it's dietary in nature. And that's why type 2 usually occurs later in life, because it takes a long time for your cells to become resistant to insulin.
Mr. Grimm,
Yes, Holden.
So I remember in the text, it said the brain requires like a constant upkeep of glucose. So does
Yes.
that mean it doesn't need to use insulin to go to the brain if it's just constantly getting it already?
Well, no, I mean, you have to realize that the two most important organs that need oxygen all the time, and if they don't get oxygen for a minute, it will kill cells, are the two cells that are being utilized most of the time. Nerve cells in your brain are constantly being utilized, and without glucose, they will die. So it's really important to ensure we have a good blood supply that was flow and supply together, a good flow of blood up to your brain all the time. And that's why we have little monitors in our carotids that are ensuring we have a constant blood flow to our brain. Because if we didn't have blood flow to our brain, those cells would die. The other cell that's constantly being utilized is the cardiac muscle tissue, and we need oxygen always going there as well. Those two cells do need insulin that allows glucose to go in at faster rates, but they also have glucose transport proteins that do allow more glucose to get in without the help of insulin than any other cell. Are you leaving me, Holden? You're like, yes, it's already like 4 minutes after the bell, man. Take care, buddy. Have a great weekend. Bye, Rohan.